You can go.
So welcome everybody and good afternoon. Thank you for attending this webinar on Duchenne muscular dystrophy genetic diagnosis organized by EURO-NMD in partnership with European Academy of Neurology and EURO-NMD for rare neurological diseases. I wish to start by thanking our today's speaker, Alessandra Ferlini, Rita Selvatici, Francesca Gualandi, and Fratter for taking the time to share their knowledge with us. So little presentations about our today's speakers. So Alessandra Ferlini is specialized in medical genetics, neurology, and holds a PhD in genetics. She's an associate professor in medical genetics and director of the medical genetics unit at the University of Ferrara in Italy. Fratter is a consultant clinical scientist and leads the mitochondrial diagnosis team at Oxford Genetic Laboratory in the UK. He has also specialized in the genetic diagnosis of mitochondrial disorders for over 20 years. Rita Selvatici is specialized in medical genetics and clinical lecturer of the University of Ferrara in Italy for the diagnosis of hereditary genetic diseases. And finally, Francesca Gualandi is specialized in medical genetics and also holds a PhD in biotechnology. She's a senior consultant geneticist and working at the unit of medical genetics at the University Hospital of Ferrara in Italy. So some little information about today's webinar. The talks have been recorded, but the speakers will participate live in the Q&A at the end of this webinar. So don't hesitate to write your questions on the Q&A box. Regarding today's webinar, it will be longer than usual, about 1.5 hours, to accommodate all the presentations that make a logical ensemble on the theme of Duchenne muscular dystrophy diagnosis with an emphasis on genetic diagnosis. So we will start with Professor Ferlini, who will give us a detailed introduction of Duchenne muscular dystrophy. Then Fratter will discuss genetic tests for dystrophinopathies. Rita Selvatici will talk next about challenges in molecular diagnosis of dystrophinopathies. And finally, Francesca Gualandi will conclude with the genetics counseling in dystrophinopathies. So again, thank you all for your participation, and let's start the webinar. So Antonio, can you unshare your screen for me to share it? Thank you.
Good afternoon, I'm Alessandra Ferlini, I'm a medical geneticist, and I will present you an overview.
Elizabeth.
As a X-linked recessive disorder.
Yes.
Please restart, because we missed some time and we need to edit the video before putting it.
Good afternoon, I'm Alessandra Ferlini, I'm a medical geneticist, and I will present you an overview about Duchenne muscle dystrophy, focusing on etiopathogenesis and pathomechanisms. Duchenne muscle dystrophy is an inherited disease as a X-linked recessive disorder, and then males are affected. Females might be completely asymptomatic carriers. Very rarely, we have a few exceptions with symptomatic or severely manifesting carriers, even a Duchenne-like phenotype. The Duchenne muscle dystrophy is a rare disease with an incidence of 1 in every 5,000 males born worldwide, and mutations in the dystrophin gene reduce or prevent completely the production of the dystrophin protein. The inheritance of the disease, so X-linked recessive, force has to pay attention to the pedigree. So we can see here a classical, X-recessive pedigree with no male-to-male transmission. Affected males do have obligate carrier daughters, and female carriers have a risk of recurrence of the disease in 50% of males born. Duchenne muscle dystrophy is a muscle dystrophy primarily involving the skeletal muscle and has some predictable and also sequential loss of muscle abilities, talking about the muscle phenotype. At birth, in the newborn and infant, we do not see obvious clinical manifestation of the disease, but invariantly we have a high level of CK in the blood, and this is something that may just switch off attention for this boy having such a very high CK values. Around 3-7 years of age, the boys present difficulties in running, and difficulty in standing up from supine and climbing stairs. And then after 7 years till early teens, the boys have a rapid decline and loss of the ambulatory capacities. We should underline that corticosteroids have dramatically changed the DMD natural history and are now considered the gold standard of DMD treatment. This slide shows dystrophin gene and protein characteristics and also reciprocity. So, comparing dystrophin gene and protein in terms of domains and gene region. Dystrophin protein is composed by 4 domains. The actin binding domain, linking the sarcomeric F-actin, as we see in the figure below here, this is dystrophin. The large rod-like domain, composed by 24 spectrin-like repeats, and the COH terminal domain. This domain, as you see in the figure, is linking several glycoproteins, which are the dystrophin-associated glycoproteins, across the sarcolemma membrane, and then, connecting the intracellular to the extracellular matrix. The protein is 427 kilodaltons and belongs to the spectrin family. The function is a dual function, anchoring and abridging, so anchoring the sarcomer through F-actin and abridging to the sarcolemma by the COH terminus and then to the extracellular matrix. The importance of dystrophin, therefore, is a sort of elastic bridge between sarcolemma and the sarcomeric contractile activity. We can recognize a dystrophin cascade in the Duchenne pathogenesis as in skeletal muscle, and therefore the absence of dystrophin, as you see here again in red as a bridging protein, causes muscle sarcolemma fragility, followed by satellite cells depletion and failure of regeneration. This is a very important aspects of muscle dystrophy, because failure of regeneration is the final cause inducing the skeletal muscle to necrosis, apoptosis, and then completely loss of myofibrils and wasting of the muscle activity. Dystrophin protein, you see, can be seen by tissue analysis, immunocytochemical analysis, as showed in these slides. You see on the left skeletal muscle and heart in transversal sections, and labeled using antibodies, anti-dystrophin, and you see here very clearly the very nice localization of the protein in green, just in the subsarcolemma side of the muscle cells and the cardiac cells. In terms of expression, muscle dystrophin protein is very strictly expressed in skeletal muscle, heart, and brain, and completely absent in terms of protein in all other tissues. From the transcriptional point of view, the situation is a little bit different. Indeed, we have transcript mostly represented in these three tissues, so muscle and brain, but also present a very low level in terms of representation in other tissues where they have some functions, but with very limited level of transcript expression. From the clinical point of view, we know we have two different phenotypes, a severe form, so Duchenne muscle dystrophy, and a milder form, the Becker muscle dystrophy. And the difference is based on the dystrophin protein, which matters in terms of phenotype correlation. Indeed, the dystrophin is completely absent in skeletal muscle of DMD. You see here a skeletal muscle, from a Duchenne boy with only a few revertant fibers that can be seen somewhere and somehow in the muscle. But also we have some protein preserved in Becker muscle dystrophy. And this is mainly due to the frame rule we will see in a while in terms of genotype-phenotype relationship. And indeed, the frame rule plays a crucial role in defining the clinical phenotype of dystrophinopathies. Any mutation types could be deletions, could be duplications or small mutations in case they preserve the frame, so the way in which the gene and the transcript is translated into the dystrophin protein normally cause a milder phenotype. So frame deletion are normally associated to a Becker muscle dystrophy or even more milder phenotypes. While we, in case of out-of-frame mutations, deletions, for example, or any small mutation types in the frame shifting, so disrupting the frame of the gene, we will have a severe phenotype. The frame maintenance is therefore a rule which is followed in the vast majority of cases. Approximately 97% of cases follow the frame rule. There are exceptions, however, and both, of course, in BMD, which have out-of-frame mutations, or DMD, which may have in-frame mutations. These exceptions are, most frequently located in very specific gene regions, basically in both cases. So at the 5' of the gene, involving mainly exons 3 to 7, 9, or in the central region of the gene corresponding to the rod-shaped domain around in exon 50, so ranging from exon 44 to 52. So basically we have to pay attention in all those cases when mutations any type may occur in these two specific regions in order to check if we can apply an exception to the reading frame rule. Duchenne is a muscle dystrophy, classically, classified according, of course, to the neuromuscle disease classification, but we are changing a little bit now our view because we know more about gene function and protein function and their expression sites. So Duchenne is indeed a multiple diseases, as we say here, because invariably Duchenne boys and many Becker boys do have a dilated cardiomyopathy with variable onset. It might be early or late onset, and we can also recognize a very specific phenotype called X-linked dilated cardiomyopathy, which may also occur without any muscle symptom, very rarely, of course, but is very well known, this phenotype. We can also recognize that mutations occurring in some specific regions, for example, around exons 29 or 45, 49, or around exon again 45, 52, 55, so basically in the rho domain, may frequently develop a dilated cardiomyopathy. The same we may see for intellectual disability, which is very frequently occurring in Duchenne muscle dystrophy boys, which is strongly related to the presence or maintenance of or loss of the short dystrophin isoform, the DP140 especially, and DP71, which, absence is strongly correlated with intellectual disability or even autism spectrum disorders. And also epilepsy is also rarely may occur in Duchenne boys, 70% of cases, again recognizing a focus on around exon 44 to 63. Dystrophin gene is finally regulated. One example is the occurrence of many alternative splicing events. In the dystrophin transcript, we have a very variable exon composition. Some exons are even spliced in a muscle-specific way. This is published, for example, in the quadriceps muscle, while invariably exon 4 is missing. This is just an example. And also there are events related to the splicing which have been described. I reported a very nice paper published by colleagues at UMC, where splicing is not sequential, and we have the so-called recursive splicing. So basically groups of exons are spliced together in a different timing. So this is, of course, is a very interesting event, a very interesting process, possibly affecting also deletion duplication consequences, also exon skipping propensity when we have the novel therapeutical approach of exon skipping, is isoform transcription, and definitively protein diversity in terms of translating a dystrophin protein with a different exon composition. Also very interesting is the presence of several long non-coding RNAs within the DMD locus. As you see here, this is the long mRNA of the DMD and the entire locus, and you see here, a plethora of non-coding RNAs, short RNAs, which have only regulatory functions. They do not encode for any protein, and they have a very important, gene expression role, inducing transcriptional interference, chromatin remodeling. They can regulate also alternative splicing, and also they can generate small interfering RNAs impacting on the expression of other genes. They can also modulate the rate of protein translation. They have many functions related to the cell, various cell processes and transcription processes. So the presence of these RNAs is known, but their role, their functions at the moment are poorly explored, but definitely they will have, and they, have already impact in the global regulation of the DMD gene transcription and translation process. And finally, just a very quick reminder that the DMD gene is an oncosuppressor gene. So it acts as a somatic level in cancer cells. It is very well known that this gene show somatic deletions or other mutation types in muscle-related tumors, as sarcomas, but not only. There are recent papers describing DMD Mutations, again at the somatic level, like deletions, mainly involving also the region of the DP71, which occur in glioblastoma, neuroblastoma, in meningioma, and breast cancer. So that's a very interesting function of the DMD gene that should be, kept in mind when we consider the full cohort of the gene function and the gene characteristics. And final slide, that's just to keep in mind what we have discussed, what I have showed to you, that DMD is the larger human gene locus encoding for the dystrophin protein, which is a subsarcolemma protein at the sarcolemma membrane. Transcription is finely regulated through different isoforms via alternative splicing events, and also encoding several intragenic small regulatory RNAs. The function of the protein is multiple functions, so force transducing, signaling function, structural function being part of the sarcolemma, so a structural component, developmental functions, and, as we have seen, oncogenetics function. The frame rule is still valid today and predicts severe phenotypes as Duchenne, and it applies up to 93% of cases. But of course, we have to remember that some exceptions to the rule may exist. And when we see a Duchenne boy, we should remember, we should pay attention to the presence, the possible co-occurrence of different phenotypes, the dilated cardiomyopathy, the cognitive impairment, and others, which may occur in the boys, causing comorbidities and making the DMD a multiple disease and not only a pure muscle dystrophy. Thank you very much for your attention, and this is my group. Of course, my acknowledgements are due to all people working with me in the neuromuscle diseases at the Medical Genetic Unit in Ferrara. Thank you.
Thank you to Alessandra and the Neuromuscular Diseases ERN for inviting me to talk at this dystrophinopathies workshop this afternoon. I'm going to talk about genetic testing for dystrophinopathies, and just to say that I'm a clinical scientist based in Oxford in the UK. So just an overview of what I'm going to cover in this talk. I'll start by introducing the importance of making a genetic diagnosis for dystrophinopathy. Then I will briefly summarize the DMD gene mutation spectrum. And then I will go through some key points in the European guidelines for genetic testing that were published in 2020, specifically the diagnostic genetic testing pathway, testing methodologies and technological advances of the last decade, interpretation and reporting, and finally incidental findings, for which I will include a few interesting examples at the end of the talk. The importance of making a genetic diagnosis. So making a genetic diagnosis enables confirmation of the clinical diagnosis, appropriate care, including access to personalized treatments, carrier identification in relatives, and also family planning. This pie chart gives a summary of the spectrum of DMD gene pathogenic variants or mutations. So you can see that deletions and duplications, so this is deletions of one or more exons of the DMD gene and duplications of one or more exon of the DMD gene, which together can be called CNVs for copy number variations, account for the majority of pathogenic variants, and particularly deletions 68%, duplications 10%. Then the remainder 22% of pathogenic variants are different small variants, nonsense variants, frame shift variants, splicing variants, and then extremely rare in the DMD gene, missense variants. This table summarizes the same data again really for dystrophinopathies combined here, and then here it's broken down into the Duchenne muscular dystrophy and Becker muscular dystrophy phenotypes. So just to point out really that copy number variations, deletions and duplications of one or more exon are more common in Becker muscular dystrophy and account for a higher proportion of Becker muscular dystrophy than Duchenne. I won't go through the rest of these numbers in any more detail, but these are also summarized in the European best practice guidelines that I'm going to come on to next. So in 2020, these European guidelines for genetic testing were published in the European Journal of Human Genetics. Previous guidelines were published in 2010, so updated guidelines were needed due to new genetic technologies in diagnostics and the availability of new personalized drugs. Therefore, a workshop was held in 2019, and this is a picture of the group of people who assembled in London then to start that process. The outcome of that was that the team drafted updated guidelines, and these were disseminated for consultation amongst 135 laboratories in Europe. Comments were received, and these were appraised by the authors. So the consensus guidelines were updated in light of those comments, and the guidelines were completed. They were then submitted to EJHG, and then revised and accepted, and finally published on the 18th of May 2020. So some of the key updates in these guidelines are to include next generation sequencing in diagnostics for dystrophinopathies, non-invasive prenatal diagnosis, pre-implantation genetic diagnosis, guidance on variant annotation and interpretation, a section on atypical findings, a short section on personalized medicine and clinical trials, and finally a section on incidental findings. So I'm going to go through the diagnostic testing pathway now. On the left here is the diagnostic algorithm from the publication. That's clearly too small to read, so on the right hand side of the slide I will summarize the key points. If a patient is referred with suspected dystrophinopathy, we recommend that you begin with what we've called level 1 testing to test for deletions and duplications of one or more exon of the DMD gene, the copy number variations, which account for about 78% of mutations in dystrophinopathies. If that testing is negative and there's a strong clinical suspicion of dystrophinopathy, then testing can proceed to level 2 testing, sequencing of the DMD gene for small variants, and this will account for the vast majority of the remaining causative mutations. If that is still negative and there's a strong clinical suspicion, then we recommend a muscle biopsy for histopathology. If a dystrophinopathy is confirmed on muscle biopsy, then our level 3 testing is to undertake RNA analysis of the DMD gene in muscle, and this should identify the very few remaining cases. So how has genetic testing of the DMD gene, genetic testing for dystrophinopathies, evolved over the years? So the DMD gene was first identified back in 1986, and soon after that, testing by southern blotting in the first instance, and then simple PCR and agarose gel electrophoresis followed. These are just some southern blotting results and an image of an agarose gel electrophoresis apparatus. From 2002 onwards, MLPA, multiplex ligation-dependent probe application, became a key methodology for detecting whole exon deletions and duplications. And then from the last decade, really, next-generation sequencing has become increasingly important. So just to go through the testing in a little bit more detail, so MLPA for level 1 testing, this allows quantitative detection of copy number variations. So as I've said, deletions and duplications of one or more exon. This is an illustrated example of some results in a male patient with exons 8, 9 and 10 deleted. So normally there would be additional peaks here in blue. There's no peak, no amplification of the probe for these 3 exons, illustrating that those exons are deleted in the patient. And this profile is converted by some analysis into a more statistical result here, showing that the copy number is as expected for the other exons of the gene. Then when we get to exons 10, 9 and 8, there is no product and 0 copies of the gene. of the exon. Apologies. For MLPA analysis, most laboratories use commercial kits manufactured by MRC Holland. And just to comment that capillary electrophoresis is a key step in the laboratory in the MLPA process. There are some limitations to MLPA analysis. And I'll summarize the key points here, the key limitations here. There's only one probe per exon of the DMD gene in the commercial kits provided by MRC Holland, and therefore we recommend that additional testing is undertaken if only one exon is predicted to be deleted or duplicated, so additional testing to confirm the finding. MLPA does not determine the breakpoints of a deletion or duplication, and finally, for duplications, there's no information on the location or orientation. Level 2 testing, generally by next-generation sequencing, so this is a rapid and cost-effective sequencing method for large genomic regions that makes it ideal for the DMD gene. Sequencing and/or analysis is typically limited to the coding region, so that's the 79 exons of the DMD gene and exon intron boundaries. So this is now routinely used for small variant detection in the DMD gene. And to note that it's increasingly becoming possible to undertake a simultaneous CNV detection, so the deletions and duplications of one or more exon, which can enable combined level 1 and level 2 testing in a single step. So this isn't yet routine practice in most laboratories, but this is likely to become increasingly become routine practice as we move forward. And just to note that short and/or long-read whole genome sequencing may soon become the preferred method for detection of both CNVs and small variants. And the kind of technologies we're talking about here, an example is Illumina. One of their highest capacity instruments is the NovaSeq for short-read sequencing. And then there are various third-generation sequencing, long-read sequencing methodologies that include Oxford Nanopore. So finally, the level 3 testing, RNA analysis. Muscle mRNA is strongly preferred for analysis. So alternatives such as extracellular RNA from urine may be possible, but blood RNA is not generally recommended as there's only a low level of illegitimate transcription occurring in blood. There are two main uses for RNA analysis. So rare cases where a genetic diagnosis is not confirmed by level 1 or level 2 testing, as I showed in the diagnostic algorithm earlier. So this could be complex rearrangements, deep intronic variants. They wouldn't be detected in the routine level 1 and level 2 genomic DNA analysis. Also, RNA analysis can be very useful to determine the consequences at the mRNA level of a genomic DNA variant of uncertain significance, or in cases where the genotype doesn't match the phenotype. Another key section of the guidelines is regarding interpretation and reporting. This section includes information on variant annotation. So we recommend that the HGVS guidelines for sequence variant nomenclature are used for the description of small sequence variants in the DMD gene. However, for exonic deletions and duplications, so deletions and duplications of one or more exon, the CNVs, we recommend a description in words, such as, for example, deletion of exons 45 to 50 of the DMD gene. This is because the HGVS variant nomenclature guidelines descriptions are complicated and not particularly user-friendly, so it's much more easy to understand the meaning of the result if it's described in words stating the exons deleted or duplicated. We also provide information on variant interpretation in the guidelines, which is essential for correct interpretation of genetics results, so when variants are detected, correctly interpreting the clinical significance of them. So the ACMG AMP guidelines, published by Richards et al. In 2011, form the basis of international standardization in variant interpretation. So we recommend that these and subsequent clarifications and additions to them are used for variant interpretation. The guidelines also include information on reporting recommendations. So this is for key points and statements to include in diagnostic genetics reports for dystrophinopathies. I'm not going to go through this in any detail now, but this information is available in the guidelines and can be referred to there. So I'm just going to talk for a few minutes at the end of this to close out my talk on incidental findings. So these are variants, particularly CNVs, so deletions and duplications of one or more exon in the DMD gene, that are increasingly being identified as incidental findings during other genetic investigations, for example in prenatal and pediatric microarray testing. Now, the prior probability of a DMD gene variant being pathogenic and having high penetrance and expressivity is much lower in these scenarios than when a dystrophinopathy is clinically suspected. So therefore, careful clinical interpretation is required. There are several cases in the literature of asymptomatic adult males with deletions or duplications of one or more exon. These are likely to be underreported. So this is essentially the risk here. We might detect one of these rare deletions or duplications of one or more exon that don't seem to be causing a dystrophinopathy phenotype, maybe causing a very mild dystrophinopathy phenotype. So a few examples from our laboratory that are yet to be published. So an incidental DMD exon 42 duplication. In this case, there was a male pregnancy referred to the laboratory and tested by array CGH because of abnormal ultrasound findings. This analysis identified a duplication of exon 42 of the DMD gene, which was subsequently confirmed by MLPA. This is an in-frame duplication, and so normally we would predict a Becker muscular dystrophy phenotype. Here is the pedigree. So this is the male pregnancy found to have this duplication of exon 42. So we did some subsequent family testing, found that the mother carried the duplication, grandfather did not. We were able to test a half-brother who also had the duplication. Now this half-brother of the mother is a 49-year-old male with no muscular weakness, regularly works out at the gym, so fit and healthy adult male, and his CK was within the normal range at 176. So here, this exon 42 duplication does not seem to be causing a significant dystrophinopathy phenotype in this adult male. In this case, we carried out further characterization of the duplication. We first undertook long-range PCR and sequencing of the breakpoints in genomic DNA, and this demonstrated that it is a tandem duplication of exon 42, as illustrated in the diagram here. We proceeded to RNA analysis in blood. There was no muscle sample available, so we were unable to carry out RNA analysis in the preferred tissue type. However, the analysis in blood mRNA was able to demonstrate that we had a duplication of exon 42 transcribed into the mRNA. So this corresponds to the larger fragment here, because we're getting transcription here to include 2 copies of exon 42 rather than the normal situation as illustrated here, which generates a smaller product. Okay, so that was interesting. We don't really fully understand why that's not causing a dystrophinopathy phenotype, but these are the examples that we're seeing. And a couple of additional examples from our laboratory, an exon 48 to 53 deletion. So here the deletion was identified by a prenatal array again in the pregnancy. This is a male pregnancy again. Mother was a carrier, but grandfather was found to have the deletion. And grandfather was a 63-year-old male with no muscle weakness or symptoms, and just a borderline raised CK, and normal cardiac investigations as well. So again, we've got reduced penetrance. Essentially, this deletion does not seem to be causing a significant phenotype in this male. And one final example, an exon 3 to 9 deletion that was detected in a pediatric array in this girl. So this looked like carrier status for a dystrophinopathy and hence implications for relatives, but we tested father, found to have the deletion, father's brother, and also the grandmother was found to be a carrier. Unfortunately, there was limited information available on the father, but the father's brother was a 34-year-old male with no clinical evidence of dystrophinopathy. He does have a raised CK, and the brother's CK, so that the father of the proband here was said to have a normal CK, but with no further information available, unfortunately. But again, clearly minimal symptoms associated, it seems, in these two males with this exon 3 to 9 deletion of the DMD gene. So just to summarize then, the EMQN best practice guidelines, or European best practice guidelines, provide detailed guidance for the testing laboratories. The pathway for testing generally involves testing for the copy number variations first, and then the small variants, although this can be combined into a single step, and RNA analysis of muscle is helpful for rare and/or complex cases. Incidental detection of DMD gene variants. Be aware of over-interpreting the clinical significance of these variants, as I've tried to illustrate in the examples. Testing unaffected male relatives can be very helpful to clarify the pathogenicity, penetrance, and expressivity, as I've shown. Finally, I'd like to acknowledge some of my colleagues who have been involved in this work. The co-authors of the European guidelines, Raymond, Stephanie, Rosario, Steve, Sylvie, and Alessandra, and my colleagues in Oxford, scientists Jesse and Penny, and Stephen Brady, the consultant neurologist. Thank you very much for listening to this talk. That concludes this part of the workshop.
Hello everyone. Topics of the presentation will be DMD gene, the NGS revolution, omics approach, open issues, and final reflections. The dystrophin gene is located on the X chromosome in position p21.1 and comprises more than 2.2 million base pairs, of which less than 1% is coding. 7 different promoters located upstream and within the gene drive the transcription of alternative isoforms. 3 promoters near the 5' end of the gene promote the expression of the full isoforms, which are the muscle, brain, and Purkinje isoforms. 4 other promoters located in intronic regions of the DMD gene give rise to short isoforms and therefore to truncated proteins. The short isoforms lack the actin binding terminal but retain the cysteine-rich and carboxy-terminus domains that contain the binding sites for the DGC complex proteins. Next-generation sequencing have greatly improved our capacity to identify disease-causing mutations with important benefits for research and prevention through the genetic counseling of patients' families. NGS is a technology that offers ultra high throughput, scalability, and speed. NGS can be used for a wide array of applications such as whole genome, whole exome, and targeted panel sequencing, offering several advantages such as the sequencing of millions of nucleotides, high throughput and many samples contemporarily analyzed, high test accuracy ensured by high coverage, accurately identified mosaicisms, identification of digenic inheritance or mutation load. However, NGS still has challenges to overcome. The first challenge is the output dimension, which ranges from 140 gigabases per sample in the whole genome to less than 1 gigabases per sample in the target panels. Whatever the approach, a lot of dry lab workload needs to be performed, and incidental findings are sometimes highlighted. The second challenge is variant interpretation. For this, the American College of Medical Genetics developed guidelines for the interpretation of sequence variants that are based on population frequency, bioinformatic prediction tools, functional tools, segregation and allelic tools. Based on these predictors, the variants are classified into 5 classes: AE benign, likely benign, variants of uncertain significance, likely pathogenic, and pathogenic. The third challenge is improving CMVs and dynamic mutations detections. The complexity of data processing depends on the giga output of the sequencing techniques, and the skill of a geneticist could not be enough to juggle this amount of data. So experts like bioinformaticians became essential for the genome and exome sequencing analysis, for CMV's calling, and in consideration of customized on-the-cloud pipelines, where the features of genetics is moving. Why genetic diagnosis is necessary in clinical practice of NMDs? An accurate and early diagnosis is mandatory for family planning decisions and counseling of other family members. By binding the disease-causing mutation, it is possible to establish the correct implications for disease prognosis and also patients' eligibility for emerging genetic therapies. Genetic counseling is also relevant for female carriers, prenatal testing, socialization, and contact with patients' associations. The single gene approach is recommended for prenatal testing, dynamic mutations, methylation alterations, preimplantation genetic diagnosis, and familiar mutation testing. So the single gene approach includes PCR, sequencing, MLPA or real-time PCR, and identifies an average number of 20 variants with a reporting time of approximately 5 to 10 days for the identification of small variations, CMVs, and dynamic mutations. The gene panel approach is used for known disease for which a variable number of genes are selected. Analysis of the output identifies 30 to 1,000 variants, but CMVs and dynamic mutation remain unsolved. If you look at the whole exome sequencing, we see that the output analysis can identify up to 30,000 variants with a delivery time report of approximately 3 months. However, even with this approach, CMVs and dynamic mutations remain unsolved. Whole exome analysis is recommended for non-disease genes for gene discovery when the analysis is performed in a family of 4 or in trios, for the identification of small variations when high genetic heterogeneity is suspected, and for undiagnosed cases. Whole genome sequencing can be considered the all-in-one approach if we talk about long-read whole genome sequencing. In this case, the analysis of the output can highlight up to 3 million variants by recognizing both small variant CMVs and dynamic mutations, but report turnaround timing is about 6 months. This should be the preferred approach for all variation types, and in particular when there is high genetic heterogeneity, ultra-rare diseases, atypical mutations, and undiagnosed cases. Even though NGS techniques allow rapid and comprehensive identification of DMD gene mutations, muscle biopsy is often required if no mutation is detected by DNA-based techniques, if there is no genotype-phenotype correlation, and in case of complex phenotype or other atypical features. Therefore, muscle tissue or myogenic cells are needed for transcriptome profiling to disclose RNA pathways or other signatures that characterize the DMD phenotype. For this purpose, we have set up a high-throughput system for RNA analysis of the DMD gene, which is a custom microfluidic exome assay called FluidDMD. This accurate and rapid RNA method, which was developed in our lab, has also been used successfully for urine-derived stem cells. The transcriptional profile of urine-derived stem cells was investigated by using RNA sequencing, and USCs demonstrated to be a very interesting tool for many gene functions exploration and genomic variation validation of NMD diseases. USCs have the advantage of being inexpensive, easy, non-invasive, without surgical procedures, and they can be obtained from people of different ages, sexes, and health status. This in vitro model helped us to solve several DMD patients. Here is a recent case where RNA analysis on USCs addressed the analysis to solve the following case. The patient is an 11-year-old boy from healthy non-consanguineous parents. His muscle biopsy showed a dystrophic pattern. RNA analysis of the patient was performed on urinary stem cells by using the FluidDMD card as shown at the top left. The assay highlights the absence of amplification of regions spanning exons 1 to 54 and with only the DP71 isoform expressed. Whole genome sequencing was performed and accurate analysis identified a large inversion of 15 megabases. The two breakpoints were highlighted using integrative genome visualization as shown at the bottom left that were localized in XP region 22.2 corresponding to intron 1 of the GRPR gene and in XP 22.1 corresponding to intron 54 of the DMD gene respectively. Amplification and sequencing of the two breakpoint junction shown at right identified two fusion transcripts, one driven by the GRPR promoter leading to exon 1 being adjacent to the inverted DMD exon 55 and the second driven by the MDMD promoter until exon 54, which became adjacent to the inverted GRPR exon 2. So, RNA analysis is recommended when a patient with suspected DMD-BMD has a negative MLPA, negative CGH, and sequencing results. When the clinical phenotype is not as expected, so the exception of the reading frame rule has to be checked. To check the splicing impact after finding undescribed variations, to verify genetic prognosis at the early disease stage and CK level correlation. An open question is whether RNA-seq will become a tool to identify all types of DMD mutation. Into the clinical practice, there are also open issues that concern precise phenotyping to access to more appropriate analysis, problems with variant interpretation if not described, or with mutations such as CMVs and dynamic mutation that are not covered. The diagnostic precision that should be in full, burden of storing and managing data in the post-test phase for any subsequent re-evaluation, ethical questions if we find incidental findings, excessive costs for genomic tests, lengthy turnaround reporting timing. Therefore, it is recommended robust and structured interaction between clinicians and medical geneticists, evaluating omics infrastructures, designing of collaborative figures, and sharing best practices. Thank you for the attention.
Genetic counseling is a complex process, and different definitions have been proposed over time. But the main aim of genetic counseling is to help people understand and manage the personal and familial implications of genetic contributions to disease. The standard setting for genetic counseling consists of an on-site visit where the genetic counselor and the patient physically meet face-to-face in dedicated clinics. Telegenetics is an application of telemedicine for the delivery of genetic counseling and genetic testing at distance. This approach, that has been more widely applied in the United States and North of Europe, represents an attractive alternative to traditional on-site genetic counseling, and this especially considering that genetic services are often not capillarily distributed among countries or among regions within a country, and that patients have to move sometimes long distances to reach those services. Furthermore, the rapid development of advanced and complex techniques for genomic analysis requires high-level expertise that is most often centralized. The COVID-19 pandemic has obviously further implemented telemedicine in general, including genetic counseling. As a pilot experience in Italy, in 2019 we have launched a project we have called TeleNewCare, which was aimed at evaluating the effectiveness of genetic counseling provided in remote modality for patients and families affected by neuromuscular or cardiac inherited diseases. The project involved our unit in Ferrara as providers of genetic counseling and as collaborators the Italian Neuromuscular Omni Centers, NEMO Centers, in Milan, Rome and Messina, which are specialized institutes for the care of neuromuscular patients, as well as different Italian cardiology units. In this pilot project, we have enrolled a group of adult patients affected by neuromuscular or cardiac diseases and age and disease matched controls. We have collected questionnaires, a clinical genetic satisfaction questionnaire and a telemedicine specific questionnaire in order to assess satisfaction and gather criticism for the online counseling when compared to standard in-person setting. Patient answers to items in the telemedicine questionnaires revealed an overall satisfaction for the remote counseling, and especially successful and appreciated by patients was the possibility to have a simultaneous and joint evaluation by different specialists, which is quite easy through a virtual connection, but is very difficult to organize in on-site standard clinic. Given the appreciation expressed by NMD patients for telecounseling, the activity now continues with the support of Sarepta Therapeutics in a novel service-oriented project aimed at offering the Italian network of clinicians involved in neuromuscular diseases a genetic counseling support for the interpretation of complex genetic tests and also for providing to patients personalized therapy information. Regardless of the setting, either on-site or virtual, genetic counseling includes several steps. A correct diagnosis is crucial. These steps is based on accurate collection of proband and family medical history, proper examination and investigations. Genetic counselors use a very powerful instrument to collect family and also personal history that is based on the construction and analysis of the pedigree, a graphical representation of the patient family composition. Based on the specific diagnosis and the definition of the mode of inheritance, it is then possible to estimate the risks of developing transmitting the disease and to communicate to the families the information on the medical diagnosis and its implications in terms of prognosis and possible treatments. The choice or option available for dealing with the risk management are discussed with the patients, and a long-term support is always offered by maintaining contacts with families. X-linked recessive inheritance refers to genetic conditions associated with mutations in genes on the X chromosome, as the dystrophy gene. In X-linked recessive diseases, males carrying a single X chromosome are generally affected, and females who have 2 copies of the X chromosome are healthy carriers. Nevertheless, females carrying X-linked gene mutation can be affected, and the clinical expression can be variable depending on the X inactivation phenomenon. X inactivation is a dosage compensation process by which one of the 2 copies of the X chromosome is inactivated in the female mammals. This prevents females from having twice as many X chromosome gene products as males. The choice of which X chromosome will be inactivated is generally random in humans. Nevertheless, sometimes a skewed inactivation occurs, and if the preferentially silenced chromosome is the one carrying the normal gene copy, the carrier female can be affected by a certain degree of the disease. Hallmark of X-linked recessive inheritance emerging from the pedigree analysis are that affected males are maternally linked, that there is no male-to-male transmission of the disease, and that healthy males do not transmit the disease. If the pedigree is big enough, with multiple affected individuals, obligate carrier females can be identified. For example, if a female is mother and sister of affected males, she is an obligate carrier of the gene mutation. About 1/3 of the time, the mutation in the dystrophin gene comes as a new change, a so-called de novo mutation. The rest of time, the DMD mutation can be passed down from mother to son. Germline or gonadal mosaicism is a type of genetic mosaicism where more than one set of genetic information is found specifically within the gamete cells. Germline mosaicism is a relatively frequent mechanism of inherited diseases and provides an explanation for the inheritance pattern in cases where multiple affected offspring are born to clinically and phenotypically normal parents, or, in the case of dystrophinopathies, from non-carrier mothers. Main issue in genetic counseling of dystrophinopathies is female carrier identification due to the related risk of affected children. When the familial DMD variant is known, identified in the affected male subject, a target test can be performed. Remember that if the variant in the index case cannot be found in genomic DNA from his mother, the frequency of germline mosaicism still confers a significant recurrent risk for future children. In case when the familial pathogenic variant is unknown, Female relatives at risk of being carrier should be offered the full cohort of genetic testing through CMV analysis and sequencing, since these two approaches offer a very high sensitivity. Testing should start from the woman who has the highest prior carrier risk, ideally from an obligate carrier. If no pathogenic variant is detected by CMV analysis and sequencing, a muscle biopsy in the at-risk female might be appropriate to evaluate dystrophin expression and for RNA studies. This especially in obligate carrier female or female with ICK. If a muscle biopsy is not available, urine stem cell RNA studies might be helpful to identify the typical DMD variants. Measurement of serum CK levels may also be helpful in refining carrier risk prior to or alongside genetic testing. In very rare cases, if key family members are available and the family structure is suitable, carrier risk can be determined by tracing the inheritance of high and low risk DMD haplotypes within a family. The rare instances when this analysis may be useful include when there is very strong evidence of dystrophinopathy and no pathogenic variant is detected in the proband, or when it has not been possible to identify at the genomic level the cause of a structural change detected at the RNA level in the proband. In case of pregnancy of a carrier female, for considering prenatal testing, the familial pathogenic variant has to be ideally known in advance of testing and should be confirmed before the prenatal test. In case the pathogenic variant has not been previously identified, CMV detection and sequencing can be offered to the pregnant mother. Prenatal diagnosis of any dystrophinopathy phenotype that is clinically relevant should be limited to male pregnancy only. It is not generally considered appropriate to offer prenatal diagnosis for a female fetus, considering that at present it is not possible to predict whether a female heterozygous for a DMD pathogenic variant will manifest any signs of the disorder or not. As exceptions, prenatal testing of female fetus may be considered in some situations, as the documented familial recurrence of an X autosome translocation, a documented familial complete skewed X inactivation, or exceptionally other emotional or social motivations. Different types of prenatal testing are nowadays available for dystrophinopathies. Invasive prenatal genetic testing is preferentially performed on chorionic villus sampling in early gestation. High-quality DNA and high yield is typically obtained from CVS, and these allow a wide variety of possible testing methods, including the NGS approach. Therefore, results can be obtained and reported rapidly, normally by 12 days from sampling. Amniocentesis, typically undertaken at 15 or 17 weeks gestation, may be used in rare cases, but it has the disadvantages both of being taken later in the pregnancy and of lower DNA quality and yield from direct amniotic fluid. Therefore, amniocyte mini-culture may be required to obtain enough high-quality DNA. The non-familial DMD gene pathogenic variant can be tested using the appropriate technique that was previously used to identify the variant in the proband. A check for maternal cell contamination must be carried out, since its presence at a significant level may affect the interpretation of the fetal result. This can be achieved by the analysis of intragenic DMD microsatellite markers that will allow also confirmation of the fetal sex. Invasive procedures are associated with a 0.51% risk of miscarriage. Beyond invasive procedures, it is nowadays possible to perform prenatal detection of dystrophin mutations in a minimally invasive way. taking advantage of the presence of fetal genomic sequences circulating as free DNA in the maternal blood during pregnancy. Cell-free fetal DNA was first identified in 1997 when Y chromosome material was detected in the plasma of women carrying male fetuses. Cell-free DNA originates from trophoblastic cells of the placenta, and it is always first detectable from 4 weeks gestations. However, cell-free DNA only reaches levels that can be reliably used for clinical testing from around 6-7 weeks. It is fragmented and it is present in the maternal plasma alongside maternal cell-free DNA. The fraction of fetal cell-free DNA increases throughout the pregnancy and is clear from the maternal circulation within hours from birth, and is therefore specific to the ongoing pregnancy. The presence of cell-free DNA in the maternal circulation has enabled the introduction of non-invasive prenatal screening or testing for aneuploidy, also called NIPT, and non-invasive prenatal diagnosis for monogenic disorder, that is called NIPD. As anticipated, fetal fraction in the maternal blood can be reliably used for detecting aneuploidies. That is a variation in the number of specific chromosomes, mainly in chromosome 13, 18, and 21, associated to severe conditions. Massively parallel sequencing technique can be used, and both maternal and fetal fragments are counted and analyzed in NIPT. Sequences are aligned to genome to identify the chromosome origin of the fragments, and aneuploidies are detected by comparing the amount of chromosomal material against a set of reference chromosomes. NIPT for aneuploidy is a screening rather than a diagnostic test and requires confirmation of positive results with an invasive test. This is due to the risk of discordant results caused by confined placental mosaicism or by maternal neoplasms or shedding of fetal DNA from a vanishing twin. Differently from NIPT, non-invasive prenatal diagnosis of monogenic disorder is a diagnostic test, and it does not require an invasive test to confirm a positive result. NIPT has substantial technological challenges which have played part in the slow implementation of this diagnostic testing compared to NIPT. The fetal fraction of cell-free DNA is relatively low and commonly comprises only 5–20% of the total circulating cell-free DNA in maternal plasma. Detection techniques, therefore, need to be highly sensitive to detect low level of variant in the fetal DNA. The utilized technique can be broadly categorized into two main groups: the detection of de novo or paternally inherited pathogenic variant for dominant conditions or for recessive diseases where the mother and the father carry different mutations, and dosage-based techniques for the detection of maternally inherited variants, as in case of maternal DMD mutations. The first of this group requires low-level variant detection, as the mutation searched for in the fetus will not be present in the maternal cell-free DNA and will therefore only be found in the small fetal fraction. The second group relies on more complex dosage analysis in order to assess the ratio of wild-type to mutant alleles in the cell-free DNA in order to establish whether the fetus carries a variant that is present in the mother. To achieve this level of sensitivity, techniques such as relative mutation dosage and relative haplotype dosage have been developed. Relative mutation dosage measures the proportion of wild-type and mutant allele in cell-free DNA. The reported examples refers to a recessive disease. For homozygous affected fetus, relative mutation dosage will demonstrate increased level of the mutant allele, whereas if mother and fetus share the same genotype, allelic balance is predicted. Where the fetus is homozygous wild type, relative mutation dosage will indicate reduced level of the mutant allele and an increase in the relative wild type allele contribution. Non-invasive prenatal diagnosis for dystrophinopathies is likely to increasingly become feasible using methods such as relative haplotype dosage analysis. Relative haplotype dosage can be applied for NIPT of single gene disorders and goes beyond paternal inherited mutation to combine relative mutation dosage with linkage analysis. Relative haplotype dosage analysis measures the allelic imbalance between two haplotypes in plasma cell-free DNA to determine which haplotypes has been inherited by the fetus. Haplotype phasing is conducted through sequencing of small nucleotide polymorphisms. In the case of DMD-BMD, the male proband DNA provides the haplotype linked with the mutated dystrophin gene, that is, the affected haplotype, while the maternal DNA is needed to identify the informative heterozygous SNPs that will be used in the RHDO analysis. The allelic imbalance between the two haplotypes in the maternal cell-free DNA is used to calculate the fetal fraction, which has been set at 10% in the reported case. In the reported example, haplotype A, the affected haplotype, is overrepresented in the maternal cell-free DNA, indicating that it has been inherited by the fetus. There are several limitations of RHDO analysis. One is in the analysis of consanguineous pregnancies, where haplotypes that are identical by Duchenne may result in a lack of informative SNPs. The second is the need for DNA from an affected family member to allow for haplotype phasing, and this is not always possible. This is an example of non-invasive prenatal diagnosis application to DMD. In the family, the patient was a carrier of a deletion of exon 45 in the dystrophin gene, which is associated to Duchenne muscular dystrophy. She had a previous affected child, and she was 8 weeks and 4 days pregnant at blood draw. Capture enrichment was designed to target highly heterozygous SNPs across the dystrophin gene region to be sequenced by NGS, and genomic DNA from the affected boy was used to determine the affected haplotype. The fetal fraction of plasma cell-free DNA was measured at 9.24%, and RHDO analysis identified 23 haplotype blocks, 12 in the forward analysis and 11 in the reverse. All haplotype blocks were classified as affected, indicating that the fetus had inherited the mutated dystrophin gene from the mother. MLPA analysis on the invasively obtained chorionic villus sampling confirmed the outcome. One limitation of RHDO analysis is the need for DNA from an affected family member to allow for haplotype phasing. And this is not always possible, and for this reason several methods are being developed in order to allow diagnosis to be feasible without requiring the presence of a proband. One of these methods is called targeted locus amplification, TLA, and this strategy allows a high number of heterozygous variants from a locus of interest, for example the dystrophin locus, to be phased to either the disease-linked or the wild-type allele. By massive SNP sequencing of parents' DNA, TLA allows to build up allele-specific haplotypes, including the affected haplotype around the DMD gene mutation. During NIPT, cell-free DNA is isolated from maternal plasma, will be enriched for fragments originating from the DMD locus and sequenced. The parental haplotype data, along with the cell-free sequence reads, are then used to determine which haplotypes have been inherited by the fetus. Despite TLA and other direct phasing haplotyping methods have been applied in some cases in the literature for the non-invasive prenatal diagnosis of DMD, nevertheless further studies should aim to refine the methodology before full introduction in clinical practice. An alternative to prenatal diagnosis that is becoming available to deal with the reproductive risk of female carrier of dystrophin mutation is pre-implantation diagnosis. PGD or PGT for aneuploidies is a procedure used in conjunction with in vitro fertilization in which the embryo is tested for certain conditions prior to being placed in the womb of the woman. After in vitro fertilization, a small number of trough ectoderm cells can be obtained by a blastomer biopsy without compromising embryonic viability. The developing embryo can thus be tested for the inheritance of the risk allele causing a particular disorder, and only unaffected embryos can be selected for transfer to the maternal uterus. Different molecular techniques have been used from the first report on children born after PGT, published in 1990. Technique from the single-cell simplex PCR to multiplex PCR, and in the last decade to genome-wide technologies. The genome-wide methods yield data on genotyping as well as on chromosome copy number, thus allowing for concurrent analysis of aneuploidies together with the detection of mutation associated to monogenic disorders. Genetic testing of single or few cells is challenging, and the overall PGD procedure is quite complex. Thus, PGD is a specialistic test carried out in a limited number of centers. Nevertheless, it represents a well-established alternative to invasive prenatal diagnosis for several diseases, and in the field of dystrophinopathies, this type of genetic diagnosis is appropriate and feasible, and it is applicable to both CMVs and to small DMD variants. Thanks for the attention.
Thank you to all the speakers. It was a very instructive session. We have a few questions that we would like to put to yourselves. One is from someone who left the meeting in the meantime, so I will voice it. She was thinking about the established newborn screening methods, I suppose, because she was asking if there is any chance of using dried blood spots for dystrophinopathies.
Thanks, Antonio. Shall I try and answer that, or are you answering that, Ali?
I can provide a very quick answer, and then Carl, if you can add, of course, fine. So, of course, yeah, there is no, ongoing newborn screening for Duchenne. At the national health systems, as far as I know, in Europe, so not in any countries, despite of some pilot newborn screening studies have been done. Of course, is doable on DNA that we can extract, we can get from DBS, so dry blood spots. This is an interesting point, an interesting question, and obviously the point is that there are sort of large discussion about the clinical utility of newborn screening in health. Definitely, newborn screening may represent the way to get a very early diagnosis in all rare disease patients. So from this point of view, it's really wishable. There are some points, really technical points, but also ethical aspects that should be discussed, should be faced. And there are many initiatives across the world on newborn screening in Europe, in the United States, the BabySec in UK, and then Carl may add something about genomics England, and also recently approved the project from European Union, which is the Screen for Care, which I am involved in. So Carl, please add if you like to comment. Thank you.
Thanks, Ali. That sounded very comprehensive, so I'm not sure there's much to add. I guess you alluded to technical issues. So obviously a dried blood spot is not an ideal sample to get high quality DNA from. You get sort of poorer quality DNA and less of it. So that poses technical challenges to genomic and genetic analysis. But as Ali said, it is possible and feasible to work and do genetic analysis on dried blood spots, newborn screening. I'm also not aware of any ongoing programs. One of the pilots that did happen was a pilot in Wales in the UK for a number of years, but their first line test there was measurement of the CK on the dried blood spot, and genetic testing followed as a secondary test in the pathway, which probably for newborn screening for Duchenne, that would be a sensible strategy if that was being implemented. But yes, it could also be part of wider, more complex proposals for much wider newborn screening by genome sequencing, which is, as Alessandra just said, poses a lot of ethical issues and quite a complex area that requires further discussion, I think. Thanks.
Okay. So one question that is related in a way with the previous, with this one, is when do you offer? It's from Corinna Andrich. When do you offer predictive testing to an asymptomatic young sibling of an affected child?
Carla, that's for, please go.
I was thinking that was going to be for Francesca, but would that be most appropriate for you to answer from the genetic counselling side?
Also part of the guidelines, so because we were involved. So you can start, and then of course Francesca can comment. We can just be, speaking freely. Okay, no problem.
Yeah, so I guess any of us can start answering that. So was the question, when is it appropriate to do? So are we talking about in a male?
Yes.
Yeah. So you would do that at any age, wouldn't you? Because the potential benefits that Alessandra was just alluding to of getting a diagnosis early with clinical trials and things like that may be available. With Francesca can talk about the appropriate counselling, but there wouldn't be any age limitation on that, I wouldn't have thought, because of the potential benefits of getting it, of knowing early. Would you agree, Francesca?
Yes, of course, because in general, genetic testing in children is appropriate when there is an advantage for the children itself. So in case of dystrophinopathy, I think that yes, testing should be offered, can be offered early, because there is a possibility to be involved in trials or to have the advance of to assess to therapies. So I agree with that.
Okay, thank you. I'm going to unmute Deepti Bhaskar. Is interested in knowing about the manifestations of DMD on the gastrointestinal system. Deepti?
Hello, I am Dr. Deepti Bhaskar from India. I'm doing my postdoctoral fellowship in neuromuscular disorders at NIMHANS. First of all, I just want to congratulate and thank all the organizers and speakers. I was just interested in the gastrointestinal manifestation of DMD. So recently in a neuromuscular clinic, came across a patient with aplasia cardia, genetically diagnosed a DMD patient. I'm just curious to know about what are the various manifestations and how common it is to see in DMD patients.
You're muted, Alessandra.
Yeah, thank you, Deepti. So I hope to got all your questions. So you know, dystrophin is expressed in smooth muscle as well. So definitely we have gastrointestinal manifestations, especially at the late stage of the disease, which are characterized by reduced intestinal motility, which causes you know, a lot of troubles in aged DMD boys. Just also to underline that intestinal muscles, so gastrointestinal traits includes both striated and smooth muscles. So it's you know, just a problem of the entire muscle body of the gastrointestinal traits. There was another, did I answer to your question?
Yes, doctor. Thank you for that.
Thank you.
Thanks.
All right, and we are going to end with a case that Natalia Kokrina indicates of a boy 4 years old, asymptomatic, CK 24,000, LMPA duplication exon 2. And she's asking what is the diagnostic option to find out if it's Duchenne or Becker.
Yeah, Carla.
I'm not totally sure if I can answer that one fully, but because is it not clear cut clinically Duchenne anyway, but I'd have to look up about exon 2 duplications. I was and because it's not in my head, off the top of my head, what's been reported previously for duplications of exon 42, because sorry, exon 2. I don't know if any of the other panelists know if that's been variably reported. Is that the reason why that there's known cases to be variation from Becker to Duchenne with that duplication? I mean, it sounds like the clinical picture sounds, with that really high CK, sounds very Duchenne-like anyway, doesn't it?
Yeah. Anyway, sorry.
No, just to add that the CK is terribly high. So I would just suggest to you know, a very, very careful follow-up for this boy, although it is asymptomatic at the moment, okay? So just to monitor the clinical picture.
Natalia, if you have anything to add, you can unmute yourself. Otherwise, I would add that if you want, we can organize a CPMS panel to discuss the case, and that would be more appropriate to have the proper detail and the proper answers provided to yourself.
Yeah, excellent idea, Antonio. Yes.
Yeah, okay. Because we haven't got all the information to hand to give a full answer now, unfortunately. And like I say, I haven't got all the previous genetic reports of that duplication to hand. Yeah. So apologies for not providing a fuller answer.
Okay, so
it's possible.
We can provide in a different format. Okay, thank you very much. It's been a long but very interesting session. I've learned a lot, but that's not new. I usually learn a lot with all our speakers. They are of very high quality. Fortunately, we have a very nice network from that point of view, and I hope to be talking to you and listening to your talks very soon. Thank you very much for your help, for your participation and your goodwill. Thank you.
Thank you very much, and thanks to all, Carla, Rita, Francesca, Antonio, and all the EURO-NMD fantastic team. Thank you so much, guys. Bye-bye.
Thank you very much.
Thank you. Yeah, thank you, everybody.
Thank you.
Thank you.
Thank you.